For the sense organ lab, which is going to cover 2 of the 5 special senses, which is going to be ear and eye. All right, so what you want to do, guys, is I'm doing this lab intro. You're going to want to kind of follow along with your lab manual. All right, so let's get started. So the first topic we're going to talk about for this particular lab is going to be the ear. All right. So if you look at the first page of your lab manual, it asks the ear has 3 regions and 2 functions. List each, right? So in terms of the 3 regions of the ear, right, there's an outer ear, there's a middle ear, and there's what's called an inner ear, right? Now overall, when you think about the function for the ear, it has 2 overall functions, and one is the obvious one, which is hearing. And the other function is equilibrium, or we commonly known as balance. All right. So the outer ear, okay, consists of this part right here, right? This is called the auricle. All right. In addition to this part right here, this is called the external auditory canal. Now, the only function, guys, of the auricle, it's going to be of the external ear. The only function is hearing. Okay. The second part of your ear is found inside in your head. Okay, it's carved into your temporal bone. We call this second portion of your ear, we call this the middle ear. Okay, and we're going to see the middle ear consists of the tympanic membrane. Okay, it consists of the tympanic cavity. It's an air-filled space inside your temporal bone. Okay, and these 3 small bones here, okay, we're going to talk about later. These are known as the ossicles. Okay, so the inner ear, okay, the only function of that is also hearing. And then basically carved into your temporal bone, okay, it's not a solid structure. We'll talk about this more in lecture, right? We have what's called the internal ear, okay? Labyrinth, it means maze, okay? It's basically a series of passageways that are carved into your temporal bone. Now, in terms of function, your inner ear actually has 2 functions. Okay, it functions both in hearing, therefore the receptors for hearing are located somewhere in here, and also for equilibrium. Okay, and the receptors for equilibrium are also found within the inner ear. All right, so again, there's the external ear, functions only hearing, the middle ear, again, function only hearing, and then we have the internal ear, which functions both in hearing and equilibrium. All right, so start off first with the external ear, okay, and the 3 structures, right, well, really 2 structures here that we're going to concentrate on. This is kind of the boundary between the external and the middle ear, okay, are the auricle, external acoustic meatus, and again, the boundary between the 2, which is the tympanic membrane. Again, the function only for this is going to be for hearing. All right, so if you ask a child where their ear is, they're probably going to pull and tug on this part, okay, which is what we call the auricle. Okay, and if you look at it, it has a funnel shaped to it, basically. Okay, and its function is to collect sound waves and funnel them into the external auditory canal, right? So as sound waves come toward your head, right, they're going to strike different portions of your auricle, and they're going to take those sound waves then coming from different, okay, positions, and they're going to funnel them into this little tiny passageway called the external auditory or the external acoustic meatus, right? So if you ever had someone saying something really quietly to you, or there was a really soft sound, you couldn't really hear it, you probably cupped your ear, all right? And the reason why you were cupping your ears is you're actually trying to make your auricle a lot bigger, trying to capture more sound waves to funnel into your external auditory canal or the external acoustic meatus. All right. The next one, again, is the external acoustic meatus, aka the external auditory canal. It's a short, narrow chamber that's carved into your temporal bone. Okay, it's going to be lined with skin. There's going to be hairs. Okay, you're also going to have what's called ceruminous glands. Okay, and these ones here are going to secrete basically earwax, right? Now, the function for this that you guys have to know for your lab exam, okay, is that it transmits sound waves from the auricle to the tympanic membrane. Okay, transmit sound waves from the auricle to the tympanic membrane. All right, so the tympanic membrane, okay, again, it's kind of the boundary between the external and the middle ear. Okay, it's this cone-shaped structure right here. Okay, and you can kind of picture as the sound waves, okay, are traveling, okay, past your auricle, okay, into your external auditory canal, it then will strike, those sound waves will now strike your tympanic membrane, okay, causing your tympanic membrane to vibrate, okay, at the same relative frequency as the sound waves that are striking it. Okay, again, it's the boundary that separates, again, the outer ear from the middle ear. So the function for the tympanic membrane is that it converts sound waves into vibrations. Okay, converts sound waves into vibrations. All right. So what would these look like on your lab exam? What would these look like on a model that we actually have in lab? All right, so here's one of the 3 models. There's 3 of these. And right here, well, if we put a sticker here, we put an arrow here, right? This here would be considered the auricle. All right, so again, the function for that is that it captures and funnels sound waves, okay, into the external auditory canal. Okay, so this part right here, this is the external auditory canal. Okay, and the function for that again is that it transmits sound waves from the auricle to the tympanic membrane. Okay, and then this next part right here, well, this here represents the tympanic membrane on our model. Okay, and the function for this tympanic membrane again is that it converts sound waves into vibrations. Okay, it converts sound waves into vibrations. All right, so let's go to the next part, which is going to be the middle ear. Okay, so the middle ear again is this little air-filled space that's carved into your temporal bone. So you see this right here, all this right here, this is your temporal bone right here. Okay, and it's a little air-filled cavity, okay, or space carved into your temporal bone. We call that the tympanic cavity. Again, the only function for the middle ear, like the outer external ear, the only function for the middle ear again is going to be hearing. All right. Now, sound waves are coming in. Sound waves are now striking your tympanic membrane, your eardrum, causing your tympanic membrane to vibrate. Okay. Now, the dilemma here is this. Okay. The receptors for hearing, they're found all the way over here in the inner ear. Okay. So how do I get vibrations that are being created, right, at the tympanic membrane? How do I get that to the receptors that are found all the way over here? Okay, you do have to span the tympanic cavity. You have to span this little air-filled space. Okay, to span that tympanic cavity, to span that air-filled space, we're going to use the 3 smallest bones of the human body. Okay, we call these 3 smallest bones of the human body, these are known as the ossicles. Okay, so starting off from, okay, lateral to medial, right, we have what's called the malleus, okay, shaped like a hammer. We have the incus, okay, which is shaped like an anvil. And then we have one that's shaped like a stirrup right here. This is called the stapes, right? So what's going to happen is that the sound waves will strike the tympanic membrane, causing the tympanic membrane to vibrate. Articulating with the tympanic membrane is the malleus. So if the tympanic membrane vibrates, well, then the malleus vibrates, okay? Now, articulating with the malleus is the incus. So if the malleus vibrates, well, then the incus vibrates. Okay, so we're basically 2/3 the way across the tympanic cavity. Well, articulating with the incus is the stapes. So if the incus vibrates, well, then the stapes begins to vibrate. Okay, and by basically having these 3 small ossicles, we've now spanned that tympanic cavity. Right now, deep to the stapes, right, is a little window here. Right, you can't really see it on the model, but if we put a pointer here and we say deep, okay, well, then we're referring to the oval window. Okay, so if the stapes vibrates, well, then deep to that is the oval window. Well, then the oval window vibrates, causing then the fluid within your inner ear to be displaced. Okay, thereby the cells, we call them hair cells, okay, they are able then to, okay, sense those vibrations or those displacements of fluid. All right, so again, the ossicles are the smallest bones of the human body. Okay, so just to give you a perspective, here's a dime, right? So the one that's shaped like a hammer, again, this is the malleus. the one that's shaped like an anvil, well, this is the incus, and the one that's shaped like a stirrup, okay, well, then this one here is the stapes. Okay, and over here is a drawing. You guys can see of how these would articulate with one another. Now, on page 2 of your lab manual, okay, you actually see, okay, these bones, right, on page 2. And we do have a little plexiglass kind of preservative, I guess you can say, or display of these 3 ossicles. So we can put an arrow at any one of these 3. You got to tell us the specific name. You can't just say ossicle. You have to give us the specific name, okay? And the functions for all of them is they transmit vibrations from the tympanic membrane to the oval window, okay? So if we point at any one of these, you do have to know them by name, malleus, incus, stapes, okay? But the function will be the same for 3, which will be what? Transmits vibrations from the tympanic membrane to the oval window, okay? All right, now, before we move on, guys, to the inner ear, okay, you can actually see that this middle ear, this tympanic cavity, there's a tube, okay, that's going to link that tympanic cavity to your throat, to your pharynx. Okay, and the name we give this tube here, well, there's 2 names, or actually 3 names. Okay, it's known as the pharyngotympanic tube. Okay, it's also known as the auditory tube, and it's also known as the eustachian tube. Right, so all 3 refer to that same structure right there, connecting your throat to your middle ear. Now, the function for this, right, the function for this pharyngotympanic tube is that it equalizes the air pressure within your middle ear. Equalizes the air pressure within your middle ear. Okay, so why do we need the air pressure in your middle ear to be somewhat equal to the pressure out here, outside your ear? Well, here's your tympanic membrane, all right? You want the pressure on both sides of the tympanic membrane to be relatively equal so that when the sound waves strike it, it's able to vibrate freely, all right? Now, let's say there was pressure built up in your middle ear. Well, then that's going to make your tympanic membrane stiff. It's not going to make it free to vibrate, right? So if sound waves were to strike this now, and this is a stiff, okay, tympanic membrane, well, it's not going to vibrate very freely then, which means that the ossicles will not vibrate very much, which means then the oval window that's deep to the stapes won't vibrate very much, which means then the fluid within your inner ear doesn't get displaced very much. Okay, and ultimately what happens is that you perceive that the sound is very soft and the sound is very muffled. All right, so think of a time in your life when, okay, for one, this is going to hurt. If there was pressure within your middle ear and it pushes out on your tympanic membrane, well, it's going to cause a little bit of pain in your ear, okay? So think about sometimes in your life where your ears hurt, right, and sound was muffled, okay? You couldn't really hear very well, right? So you probably thought about, flying. You probably thought about driving up to the mountains when you were going to go snowboarding, right? So as you go higher in elevation, okay, the pressure outside begins to drop, okay? So as you go higher in altitude, okay, atmospheric pressure goes down, okay? However, in your middle ear, okay, let's say you started off, right, you're going to go flying, you're going to go to on a vacation after all this is said and done, after this semester is over, right? So you're sitting on the runway at LAX, which is sea level, okay? Well, this is sea level pressure, and out here is sea level pressure. So right now, okay, the pressures are equal. This is vibrating freely, okay? Sound waves are hitting it. You can hear perfectly fine, right? Your ears are not hurting yet. The second that you start to now climb in altitude as your plane goes higher and higher and higher and higher and up, right? Well, the pressure here is still at LAX. This is still the pressure at sea level. Out here, though, okay, even though they try to equalize the pressure in the cabin, airplane, the cabin of the plane, the pressure outside your ear now has dropped, right? So what happens is that because there's built up pressure here, your eardrum is being pushed out, it's becoming stiff, it hurts, sound waves hit it, all right? It doesn't, right, vibrate freely. Ultimately, the sound is perceived as being muffled. So what do you have to do so that your ears don't hurt? So what do you have to do so that you can hear better? Well, you probably heard of, people pop their ears. Well, how do you pop your ears? Does the air rush out of your ear when your ears pop? Okay, some of you probably thought that. Okay, but when you pop your ears, okay, what happens is that typically this eustachian tube here, these two walls, okay, are not fused together, but they're together. So they're basically collapsed. It's when you yawn, it's when you open your jaw wide that this eustachian tube opens up. Okay, and when it opens up, well, then the pressure that's built up here rushes out of that eustachian tube, of that auditory tube, and it basically escapes down into your throat. So that popping sensation is when the air that's built up, or the pressure that's built up in your middle ear, rushes out and then escapes into your throat. Okay, so what happens now? Well, the pressure here in your middle ear and the pressure out here in your external ear are relatively equal. Well, this vibrates freely and you can hear better. All right, so that's the function of the pharyngotympanic tube. It equalizes the pressure in your middle ear. All right, so here's an actual pictures of our models. Okay, so here is the external auditory canal. Here's our tympanic membrane, right? And so this space right here, that's that tympanic cavity. This is that middle ear, okay? And you can actually see here's that eustachian tube. Here's that pharyngotympanic tube, that auditory tube right there. Equalizes the pressure in your middle ear. So if that's your tympanic membrane, well then what's this and this? Well, this is the malleus. This would actually is labeled number 1. And then right behind it is the incus, which is labeled number 2. Okay, so here's a better perspective. Here's that tympanic membrane, okay? And then here's number 1 shaped like a hammer. That's the malleus. And then number 2 here, this here represents the incus. Okay, so the function for those two right now would be transmits vibrations from the tympanic membrane to the oval window. Okay, the third ossicle you can actually see is right here. Okay, looks like a stirrup. Stirrup, like if you're a cowboy and you're going to put your boot right there, right? To see a little bit better, there it is. Okay, there's the stapes right there. All right, so that's the third ossicle. So actually, number 2 and number 3, these okay actually would articulate with one another. So that's the stapes. All right. So the function for the stapes is that it again transmits vibrations from the tympanic membrane to the oval window. Okay, or simply transmits vibrations. All right. So that is the middle ear. So the next one now is the inner ear. All right. So the function for the inner ear, not only is it a function for hearing, but we're also going to see that it also functions for equilibrium, okay, which is another way is balance. Now, one thing to note about the inner ear is that it's not a solid structure, okay? This is not something that you can just dissect and hold in your hand, for example. Okay? And the best way I can kind of explain what the inner ear is all about is that think about a mountainside, okay, a mountain. Right? And let's say there was a cave in that mountain. Could you dissect the cave out and say, here, hey, look at this cave that I brought out of this mountainside? And the reason why you can say no to that is because the cave is not a solid structure. The cave is something carved into the mountainside. Okay? So very much like a cave carved into a mountainside, the inner ear, okay, is carved into your temporal bone. Okay, so it's a series of cavities, okay, or a cavity consisting of 3 parts: semicircular canals, vestibules, and something called the cochlea. All right. So we're over here now. This is our inner ear. Okay, so here's our stapes. Our oval window would be deep to that, right? On the other side of that is your inner ear. Okay, so there's 3 basic parts of your inner ear. Okay, the first part is the one that looks like a cinnamon roll, looks like a snail shell. Okay, this is called the cochlea. Okay, and it's this part of your inner ear. This has the receptors for hearing. Okay, so this is part of the inner ear that allows you to hear. On top of the cochlea, you see this structure here. Well, these are the structures that's responsible for equilibrium. All right, so right on top of the cochlea, I see this structure. Well, this is the vestibule. Okay, and then coming off the vestibule, you see these canals here. Well, these are known as the semicircular canals. Okay, so the cochlea function for your lab exam would be contains the receptors for hearing. Vestibule, let's be very specific. Okay, So yes, it's in regards to equilibrium balance, okay, but what it senses is static equilibrium, okay. So basically the position of your head when it's kept still, okay. So this is the vestibule, okay, and then coming off of that we have the semicircular canals, okay. These ones here, okay, sense what's called rotational acceleration of the head, okay, when your head is rotating, okay, but it's generally, again, it's for balance also. So vestibule, okay, senses static equilibrium, also linear acceleration, okay, and we also have what's called the semicircular canals, okay, these sense rotational acceleration of the head. Generally speaking, again, they're involved in balance. All right, so let's say we did this, okay, so here again, we see our cochlea, here's our vestibule, here are our semicircular canals, okay. If we were to cut through that cochlea, okay, that snail shell right here, okay, you're going to see these groups of three: big chamber, small chamber in the middle, big chamber, okay, big chamber, small chamber, big chamber. All right, so let's focus on one of these groups of three: big chamber, small chamber, big. We see it bigger right there, smaller, big. The chamber in the middle, this one right here, okay, this is known as the cochlear duct. That's the cochlear duct. All right. The chamber on top, okay, this is the scala vestibuli. And the one on the bottom is called the scala tympani. A little brain fart there. All right. Scala vestibuli on top, cochlear duct in the middle, okay, scala tympani on the bottom. The one you have to know for your lab exam is the one in the middle. Okay, the one in the middle, again, is known as the cochlear duct. Okay, and you don't have to know a function for that. That'll simply be name the specific structure on the lab exam. All right. Now, on top, this is the vestibular membrane. Okay, separating the cochlear duct from the scala vestibuli. Right. You don't have to know that one, but you do have to know this one. Okay, separating the cochlear duct from the scala tympani is this membrane down here. Well, this is known as the basilar membrane. Okay, so let's say we take this part, we're going to blow this up. Okay, so here, this is where your cochlear duct would be. Here's your basilar membrane, and then your scala tympani would be down here. Okay, so we're blowing this part up right there. Okay, now when I focus on this, okay, here's my basilar membrane, and sitting right on top of the basilar membrane, you guys can see what are called the hair cells. Okay, so there's actually inner hair cells, and there's these outer hair cells here. On the lab exam, generally speaking, we'll just call these hair cells. Okay, now the function for the hair cells, more specifically the inner ones, okay, these are the receptors for hearing. Okay, so we can say name the specific cell at the pointer, okay, and function. Well, their function is that they're the receptors for hearing. All right. All right, so what does that actually look like on a slide? Okay, so here's those groups of three: big chamber on top, little chamber in the middle, big chamber on the bottom. Okay, the chamber you do have to know is the cochlear duct. Okay, and then separating the cochlear duct from the chamber in the bottom, again, you have to know this. This is the basilar membrane. That would be named specific structure on your lab exam, right? And then sitting on top of that, you see the nuclei of cells here. Well, those are the nuclei of the hair cells. Okay, and again, these are the receptors for hearing, right? Now, the rest of all this, we'll talk about this in more detail when we do the special senses lecture, all right? All right, so again, if we were to focus, okay, let's say we focused on this part right here. Okay, you guys can see here's your cochlear duct, here's your scala tympani, here's that basilar membrane, right? Here, guys, are those hair cells right there. Okay, now everything together, okay, basilar membrane, okay, hair cells, right? We call this specific, okay, thing all together, we call that the spiral organ of Corti. Okay, so for your lab exam, you have to know the cochlear duct, you have to know the hair cells, you have to know the basilar membrane, and if I were to point all this together, well, you got to know this as the spiral organ of Corti. All right. All right, so again, what would this look like in terms of our model? All right, so if I were to pull this out of the ear, we can see here is what looks like a snail shell. Well, that there represents the cochlea. Okay, function for that again is that it contains the receptors for hearing. Okay, and then on top of that in blue, right, this here represents the vestibule. Okay, this is for equilibrium. You don't want to get specific. This contains the receptors that sense static equilibrium and linear acceleration of the head. Okay, and then coming off of that, guys, you see these tubes right here. These represent the semicircular canals. Okay, so semicircular canals. Again, generally speaking, these are for equilibrium also, but more specifically, these sense rotational acceleration of the head. All right, now this one here we haven't really seen on any of the figures, but this is something you have to know on your lab exam. Okay, this is a bundle of axons. This is actually a nerve. More specifically, this is the vestibulocochlear nerve, cranial nerve number 8. All right, and that function for your lab exam is that it sends information from the ear to the brain. Okay, sends information from the ear to the brain. All right, so that's pretty much it for the ear. We're going to move on, guys, and we're going to start now talking about the eye. All right, so the eye is probably the most complex of our organs in terms of organs for our special senses. Okay, it's what we call the dominant sense. Okay, so of the 5 special senses, when we think about hearing, equilibrium, taste, smell, right, and vision, well, vision is the dominant sense. Why? Well, we look at it, 70% of all sensory receptors are in the eyes, right? 40% of your cerebral cortex, okay, the part that's responsible for processing information, right? 40% of that is involved in processing visual information. So 7% of all receptors in your eye, 40% of your cerebral cortex is involved in processing visual information. So your eye is about an inch in diameter, right? It's spherical. Okay, the anterior 1/6 of the eye, is the one that's visible. Okay, so if someone's complimenting your eyes, they're kind of making a snap judgment. They're only seeing 1/6 of it, right? So the anterior 1/6 is the visible part, and then behind that, the part that's in your orbital cavity that you can't see very much, well, then we call that the posterior 5/6 of your eye. All right, now one structure you're gonna have to know, all right, for your lab exam is the lacrimal apparatus, or it's gonna be the lacrimal gland. Okay, and the lacrimal gland is always found superior and lateral, all right, and here it is. And the function for the lacrimal gland is that it produces tears, right? So this is where your tears come from. All right, so you don't have to really know. We'll talk about this more in lecture, okay? But you do have to know that lacrimal gland right there. All right, the next structure you're gonna have to know are some extrinsic eye muscles. All right, so here's that eyeball, and we see coming off of that, right, are these 6 extrinsic eye muscles right here. Okay, and the function for them is couple. Number 1, right, is for movement. These are the ones that allow voluntary movement of the eyes. Okay, another function of these extrinsic eye muscles is that they anchor the eye in place within the orbital cavity. All right. And here are some specific functions. Again, we'll talk about this more in lecture. All right. Now, the eye has different layers, and there's actually 3 layers that we'll talk about, and the name of these layers, we're going to call tunics, right? So kind of think of Luke Skywalker, on Tatooine or something like that, right, wearing his tunic. And there's 3 different layers. One's called the fibrous, is the outermost layer. We have what's called the vascular tunic. This is the middle layer. And then we have the sensory tunic, which is the inner layer. All right, so here we see a sagittal section through an eye, and the outermost layer you can see in white here Okay, this here represents that fibrous tunic. Okay, and there's two different parts that make up the fibrous tunic. Okay, the posterior 5/6 most, so from here to here, for example. Okay, the outermost posterior 5/6, we call this the sclera. Okay, it's the white of your eye, right? And the function for the sclera, number one, it protects the eye. Number two, it's the attachment site for the extrinsic eye muscles. And number three, right, it gives shape to the eye. Okay, so number one again, it protects the eye. Number two, it's the attachment site for the extrinsic eye muscles. And then number three, right, it gives shape to the eye. All right. Now, continuous with that sclera, as you go towards the front here, okay, it kind of has a more deep, like a greener color, for example. Okay, this right here represents the cornea. Okay. So the cornea is different from the sclera in that it's transparent, right? Light is able to pass through, right? So it has a couple functions. Well, one of the functions is pretty much the same. It protects the eye, right? Number two, it transmits light. Light is able to pass through it since it's clear. And three, it bends light. Fancy word for bend means that it refracts light. So light bends or refracts as it passes through your cornea. So number one, it protects the eye. Number two, it transmits light. Number three, it bends light. So that makes up your outermost fibrous tunic. Okay, so sclera, posterior 5/6. Okay, and then we have the cornea, the anterior 1/6. All right, the middle layer, it's highlighted here in red. Okay, and this red layer here, okay, this is known as, okay, it's part of the vascular tunic, or what we call the uvea. Okay, but that red layer, what you have to know for your lab exam is that we call this the choroid coat. Okay, this is known as the choroid coat. So a couple functions. Number one, the reason why we call it vascular because it has blood vessels. Okay, so function one is that it provides blood to the eye wall. Okay. The second function for the choroid coat is that, okay, it prevents light from scattering. Okay, the inner surface of your eye is very dark. Okay, you don't want the light bouncing around everywhere. Okay, once it hits the retina, you want it basically staying in one spot. So one of the layers that is very dark is that choroid coat. Okay, so another function has melanocytes. Another function, okay, is that it prevents the light from scattering. So number one, provides blood to the eye wall. Two, prevents scattering of light within the eye. All right, the next one, okay, so as we go towards the front. So again, this is choroid coat, posterior 5/6, right? And then we get to the front, it begins to thicken right here, right? It begins to thicken right here. Well, this thickening here, well, this is known as the ciliary body. Okay, it's also, again, part of this vascular tunic. So choroid coat, posterior 5/6, and then we have what's called the ciliary body. Okay, the function for the ciliary body is that it changes the shape of the lens. Okay, so it's going to actually change the shape of the lens. So you can kind of picture if you're viewing your computer screen from very close, your lens has one shape. But if you start viewing your computer screen from far away, well, your lens has to change. Why? Because you need to focus the light on the retina, right? To do that, you need to change the shape of your lens. So your ciliary body actually has smooth muscle, okay? And that smooth muscle allows you to change the shape of your lens. All right, now extending from the ciliary body, you have these little tiny threads right here in white. Okay, and what these little tiny threads represent, these represent the suspensory ligaments. Okay, that's another structure you're gonna have to know for your lab exam. So right here in white and here in white, we call those the suspensory ligaments. Okay, function for suspensory ligaments is they anchor and hold the lens in place. Anchor and holds the lens in place. We call these the suspensory ligaments. All right, so we're not even done with the vascular tunic yet. So again, posterior 5/6 in red is the choroid coat. Okay, we go to the front here. We get to thicken. We call that the ciliary body. Right. Continuous with that, we go in front of the lens in red. What we call this the iris. Okay, this is known as the iris. So choroid coat, ciliary body, iris right there. Okay. Now, before I talk about the function of the iris, notice that the iris is incomplete. There's a hole right there. Okay, there's a hole right here. And the name we give that hole, well, that's known as the pupil. Right? That's the pupil right there. And you guys know that your pupils can get bigger in diameter. We say that your pupils dilate, right? Or your pupils can get smaller in diameter. We say that your pupils constrict. All right? So, for example, you walk into a very dark room. There's not very much light. Well, your pupils are going to get bigger. Right? They're going to dilate. Or you walk into a bright room, what's going to happen? Well, they're going to get smaller. They're going to constrict, right? So the function of the pupil is that it regulates the amount of light that enters the eye. Okay? It regulates the amount of light that enters the eye. We call that the pupil. Regulates the amount of light that enters the eye. Now, controlling the diameter of that pupil, though, okay, is the iris. All right? And the iris has smooth muscle also. Okay, and contraction or relaxation of that smooth muscle can change the diameter of the pupil. Okay, so the function for the iris is that it changes the diameter of the pupil. Okay. All right, moving on. So we talked about the outermost fibrous, which again, posterior 5/6 is sclera and cornea. Then we talked about the middle layer, which is called the vascular tunic, which is choroid coat, right? And then ciliary body. And then coming off of that are the suspensory ligaments, okay? And then going in front of that, you guys can see that here is your iris, and there's a hole right there in front of your lens. What we call that the pupil, all right? Now, before I talk about the sensory tunic, which is this one here. What's this right here? What are these suspensory ligaments holding on to? Well, they're holding on to the lens. Okay, and the lens right here, which you do have to know for your lab exam, the function of the lens is that it focuses the light on the retina. Okay, you can kind of picture light is coming here, it hits your lens bends the light, and it focuses it somewhere on the retina. Okay, so here's your lens, focuses light on the retina. All right, the innermost layer, guys, of your eye is this kind of white layer that we peeled back right there. And that white layer that we peeled back, well, we call that, well, it is called the sensory tunic, but the specific name, we call this the retina. Okay, so this white layer that's kind of covering the surface of your eye, the inner surface, we call that the retina. Okay, and the function for the retina is that it contains the photoreceptors. It contains the rods and it contains the cones. Okay, these are the two different types of photoreceptors, the receptors for light that we have in your eye. Okay, so again, you have to know the retina function contains the rods and the cones. All right. Now, your retina, okay, has a lot more rods than cones, right? Let's kind of just very briefly talk about rods and cones. Cones are active in very bright light situations, okay, and they generate very sharp colored images. Rods, on the other hand, okay, these ones here, okay, are active in very low dim light situations, and they generate kind of grainy, kind of black and white images. All right, so during the day, you're really using your cones. During very low light situations, at night, for example, you're really using those rods. All right. Which leads us to this next one here, okay? So again, the job of your lens is to focus light on the retina, okay? Now, specifically on the retina, there's a spot, okay, called the fovea centralis. Let me see it right there. Let's go to this one first, okay? It's called the macula luteae or the fovea centralis, okay? And these regions here have the highest concentration of cone photoreceptors, okay? And it's found directly behind the lens, okay? You can see it right here. Here's my fovea centralis, here's my macula luteae. Contains the highest concentration of cone photoreceptors. Well, if it has the most cones, well, what is it going to generate? Well, it's going to generate very sharp colored images. So the macula luteae and fovea centralis, this is responsible for central vision. This is responsible for generating very sharp color images. Okay, because when you look directly at something, where are you focusing it? You're focusing it right on that macula luteae. You're focusing it right on that fovea centralis. Okay, so it's responsible for central vision. Okay, it's the highest, okay, level or the greatest, okay, visual acuity, which is right there. All right, going back to this slide here. This structure coming off your eye, this is called the optic nerve. Okay, and you're gonna have to know that for your lab exam also. Okay, and the optic nerve sends visual information from the eye to the brain. Okay, sends visual information from the eye to the brain. This is the optic nerve. All right. Now the region of the retina where the optic nerve exits the eye, it doesn't have any rods. It doesn't have any cones. There's no photoreceptors in that region of the retina. Okay, so what's the consequence of that? If you were to focus light on the region where the optic nerve is exiting the eye, well, there's no rods and cones. Well, you're not going to basically sense that. So we call this region of the retina where the optic nerve exits the eye, we call this the optic disc. And because it has no photoreceptors, well, the function, guys, if we ask for it, the function we'll just say is the blind spot. Okay? The region of the retina where the optic nerve exits the eye, that is the optic disc. Function, blind spot. All right, we did that one already. Okay, so let's kind of blow it up. If we were to take this retina here and kind of give you a figure, what does that look like? For example, let's blow this one up. Okay, you can see that you have different layers of cells. Okay, so here's your choroid coat in red. Okay, there's the choroid coat. Your retina has a pigmented layer. Okay, this is a single layer of melanocytes. So this is part of your retina right there. So why do I need pigment? Well, again, you want a very dark surface. You don't want light bouncing around in your eyeball. After that, you do have your rods and cones. Okay, so the rods are the yellow ones here, a lot more numerous. And then the cones are the purple ones here, not as numerous. So again, these are the ones responsible, okay, being active, very, very bright light situations and generate colored images, sharp color images. And then these yellow rods here are, okay, functional in very low dim light situations and generate kind of grainy black and white images. All right, so light comes in here, okay, and it strikes your rods and cones. Okay, and then there's a series of reactions that occur, and we'll talk about this in physiology. But then your rods and cones, they synapse with these cells here. Well, these are your bipolar cells. Okay, and then your bipolar cells talk to these cells here in gray. Well, these gray cells here are the ganglion cells. Okay, so you have your rods and cones, you have your bipolar cells, you have your ganglion cells. And notice these axons here. Okay, what's going to happen, guys, is those axons will then merge and they're going to form, okay, the bundle of axons that forms your optic nerve. Right, so you actually can see here, okay, here is your sclera, here's your choroid coat in pink. There's that single layer of pigmented cells again. If I go back to here, single layer of pigmented cells of your retina. Okay, so we go here, you can see it here, purple. And then you have your rods and cones, you have your bipolar cells, and then you have your ganglion cells. Okay, and these ganglion cells, look at their axons. Their axons bundle up, and it's these axons of the ganglion cells that will form the optic nerve. All right, now what else about your eye? Okay, well, here's the lens again, okay? Behind the lens, this big space right here, this big space is filled with a fluid. Okay, well, that's the name of the space is called the posterior segment, right? And the posterior segment is filled with very jelly, gelatinous-like fluid called the vitreous humor. Okay, and there's going to be several functions. You do have to know that for your lab exam. But the vitreous humor does what? Well, number one, okay, it transmits light. It's clear. Light is able to pass through it. Okay. Number two, right, it gives shape to the eye. It's under pressure. It's under intraocular pressure. Okay, your eyeballs are very solid structures. They're not soggy, right? And the reason why they're firm is because of the pressure of that vitreous humor. Okay, so number one, it transmits light. Number two, it gives shape to the eye. Okay. Number three, right, is that it bends light. Light is bent as it also passes through the vitreous humor. All right. Now, if behind the lens is called the posterior segment, well, in front of the lens then, okay, so lens to iris, iris to cornea, okay, this fluid-filled space here is called the anterior segment, okay, and the name of the fluid that fills your anterior segment, we call that the aqueous humor. Okay, so this space from the cornea to the iris is filled with aqueous humor. From the iris to the lens, this space here is also filled with aqueous humor. Okay, so unlike the vitreous humor, which is not renewed in your lifetime, right? Aqueous humor is constantly renewed in your lifetime. Okay, it is a blood filtrate. All right, it's going to supply what? Nutrients to the lens. It's also going to supply nutrients to the cornea. All right, so what are these things? Everything that we talked about, okay, that you're responsible for on a model. Well, here's our model. Okay, so let's kind of take this part by part stuff that we can see. Right, so here's the eye. Okay, always superior and lateral again. This pink thing, this is a lacrimal gland. Again, the function for that is that it produces tears. And then we have all this in white. Okay, this is kind of that posterior 5/6 outermost layer here. All this white stuff you see, well, this is sclera. Okay, protects the eye, gives shape to the eye. It's also the attachment site for the extrinsic eye muscles. This is sclera, right? Now, continuous with that sclera, okay, it's not really seen on this, but there's kind of a dome, a glass. Okay, it's actually present on the other eye model. It's continuous with that, okay, which would be the cornea. Okay, on this particular one, it's not visible. Okay, it's visible on the older model. All right, what else can we see? Well, coming off the back over here, well, that would be the optic nerve. Okay, so it transmits visual information from the eye to the brain. And what else can I see here? Well, you can see these red extrinsic eye muscles. Okay, and the function for that is that one moves the eye. Well, that's under voluntary control, right? And also anchors the eye within the orbital cavity. Extrinsic eye muscles. All right, so let's open it up a little bit. Okay, so you can actually see in the sagittal section, here's that outermost posterior 5/6 in white. Well, this would be sclera. Okay, deep to that now, you guys can see in kind of a reddish color here. Well, that posterior 5/6 would be the choroid coat. So the name function for that is that provides blood to the eye wall. Okay, prevents light from scattering. Okay, as we go to the front, you guys can see it thickens right here. Okay, and this thickened part here, well, continues with the choroid coat. Well, this is the ciliary body. Okay, and the function for that is that it again changes the shape of the lens. You actually see they colored it different right there, right? So that's a smooth muscle that allows you to change the shape of the lens. Now, you can't really see it here. We're going to see it on a sheep eye, but picture suspensory ligaments extending from the ciliary body and attaching to the lens. So picture suspensory ligaments extending from the ciliary body and attaching to this clear lens, which is back here. Excuse me. All right. Now, continuous with that ciliary body, you can see this blue structure here. Well, that is the iris in blue. Again, the function for that is that it changes the diameter of the pupil. And then there's a space right there. You can see, well, that space right there represents the pupil. And the function for that is that it regulates the amount of light that enters the eye. Okay? All right. Now, in the back, okay, the innermost layer here, guys, okay, and actually you can see they peel back a triangular piece, okay, of that. So here's like it's a darker, deeper red, but here it's kind of a cloudy-ish color. The reason why is we can see it here. I'm going to peel back a triangular piece of this little thin cloudy layer. Well, that little thin milky white layer there, that represents the retina. Okay, and the function for the retina, again, is that it houses the rods and the cones, the photoreceptors. Okay, and then in this spot right here, well, this would be the optic disc. This is the blind spot. This is where your optic nerve exits the eye. Then right here, guys, you see that yellow dot right there? That little yellow dot will be the fovea centralis and the macula luteae. Right there. So this is the region of the retina that has the highest concentration of cone photoreceptors. All right. So this is responsible for what? Responsible for central vision. That is the macula luteae. All right. So here's something else you guys will have to know for your lab exam. This is a sagittal section through the eye. All right. So you can see a couple things here. This outermost layer right here, posterior 5/6, that would be the sclera. Okay, continuous with what? Anterior 1/6. Well, this would be the cornea right here. Okay. Now deep to that, you can see, okay, this kind of black layer right there, kind of spotty, right? Well, that's the choroid coat, which is continuous with what? Well, this thickened part here, or that thickened part there. Well, this is the ciliary body right there. Okay, that's the ciliary body. And it's broken off. Okay, some of our slides are a little bit better, but you extending from the ciliary body, you can actually see it broken off here and here, for example, and there's little remnants there. Okay, it used to be suspensory ligaments extending from the ciliary body and attaching to this. Well, that is the lens. Okay, so this large thing is the lens. Now, continuous with the ciliary body and going in front of the lens, well, what is this here? And what is that there? Well, that is the iris. Okay, and the iris is incomplete. There's a big space right there. Again, that is the pupil. Okay, so back here, this would be the posterior segment. This is going to be filled with vitreous humor. And then over here, this is the anterior segment. This is going to be filled with aqueous humor. Okay. Now, if you were to take your microscope and look at this region here in a lot more detail, you're going to be able to see the cells of the retina. Okay. So let's actually look at not the retina yet, but different things here. But you can see here, here's a sclera. So if I were to go here and look at this outer flaky stuff out here, well, this is all sclera out here. Okay, and then right here you can actually see, well, there's the choroid coat. Okay, so this is sclera, and there's the choroid coat right here. Okay, all right. So what if we looked at this at a higher magnification? Well, this is the cornea again. Okay, so if I did that, you can see here's the cornea right here. Okay. Now, if you look at this, you can see this coming off here and that coming off. There's a pink thing in the back. Well, if we kind of move our stage closer in that direction, well, there's that line here. There's that line right there. And here's that pink thing in the back. Well, what's this? Well, here's the iris coming from one way. The iris coming the other way. Here's that space between that. This is the pupil. Well, then this pink thing behind that, this must be the lens back here. Okay? So again, see, here's the iris coming from one way. Here's your pupil. And here's the lens back there. Okay? Again, these are pretty much the same right there. Okay? Now, what if I were to now look at this region right here? Okay? So again, this, what we saw right here, guys, is the iris, pupil, lens. Okay? So iris, pupil, and then lens. What if I looked over here, though? Well, you're going to see this ciliary body. So if I do that, you guys can see here's that ciliary body. Here are these suspensory ligaments that are coming off that are going to attach to the lens right here. Okay, so this slide here shows it a lot better. Suspensory ligaments extending and attaching to this lens. All right. So again, what you're going to want to do, guys, is look at the slides, Professor Grigorik's slides, to see the rods and cones. Also see the fovea centralis slide that's over there. Okay. Oops. All right, so let's look at a dissected sheep eye. Okay, so this is what you would have done if we had a lab, all right, on campus. But this is a sheep eye. From the outside, you can see here's the cornea, here's the sclera, and here's the optic nerve that comes off the eye. Now, what you would have done, is that you guys would have done a coronal section through the eye. Okay, so you made your cut right here. You would have separated an anterior from a posterior. Right, and let's focus on the anterior half first. Okay, so this is what it would look like. And there would have been a lot, of vitreous humor right here. Okay, so this would have popped out, right? And the lens, which would have been right here, is right there. Okay, so that's the lens. And all this gelatinous-like fluid that came out with it, well, again, that's the vitreous humor. Okay, so imagine this was here, and we popped it out, and that's what it would look like right there, right? Now, from this side, you can actually see the pupil now, right? There's the cornea right there, right? Here's your pupil. And then on this back side here, you actually can see all of these little, kind of look like little ridges here. Well, this represents the ciliary body. Okay? So again, if I were to remove this from the eye, let's say we removed it here, here's your pupil, and you actually can see the ciliary body, these little ridges in black. Okay. Now it says iris here. It's not the best way to see iris. What would you have to do is actually would have to take this and flip it over to see the other side. All right. Now the second half, the posterior half, is right here. Okay, so here's the anterior half. Here's the posterior half. You see this milky white kind of coating on top. Well, that's the retina. This beige color right here. That's the retina. This contains the rods and the cones. Okay. Now in the retina, you see this little puckering of the retina right there. Okay, it's like puckering your lips, for example. But here's a puckering of the retina. The little puckering of the retina, well, that's the optic disc. That's the blind spot. Okay, so it's simply noted as a pucker on your lab exam. All right. Now, if I were to scrape this retina out of the way, like we did over here, it's black. You actually can see some of the black right there. Okay, but on the other side of that, well, then that's the choroid coat. Okay, so move this retina out of the way, that black layer that you see right there, that would be the choroid coat. Okay. Now, this is the lens. You see these black things coming off the lens? Well, what do you think these are? These used to attach to the ciliary body, which was over here. Well, these little black things coming off the lens, well, this is the suspensory ligaments. Okay? All right, so here's the actual one that's in your lab manual. Okay, this one's better than the one we actually saw. Okay, so here's a non-dissected eye. So here you see the cornea, number 1. Okay, here you guys see number 2. The white of the eye, well, that would be the sclera. 3 coming off of the eyeball at the back would be the optic nerve, right? And then here is our section that we made. Okay, so here's the front. Again, you can see the pupil, number 5. And this black stuff that surrounds it, well, this is the choroid coat. Okay, this is the choroid coat right here. Not choroid coat, I'm sorry. The ciliary body, sorry. Okay, so here's the pupil, and here is the ciliary body. Now, if I were to take this, if I were to remove this out of the front of the eye and flip it over, well, this is the front. This is the different perspective here. Okay, so there's the pupil there, and in the front it has a brown color. And what's that? Well, that number 4 then, that represents the iris. Okay, number 4 represents the iris. Again, the lens would have been right here. Okay, there's number 11. There's the lens. Okay, so the lens, right? And then all this gelatinous like fluid that surrounds it right there, well, that's the vitreous humor. Okay. All right, number 6, you don't have to know, the oris aurata, right? But if we look at this, number 1, well, that's the cornea, and then the white of the eye here, again, that's sclera. Okay. So this is the posterior half, okay? So here the retina is still attached, okay? So you can see the retina is this milky brown stuff right here. Number 10 is the retina, right? And number 8, you can see it's kind of pushed back a little bit. Number 8, that would be the choroid coat, okay? Now it's not labeled here, but see the puckering of the retina? What would that puckering of the retina represent again? Well, that puckering of the retina would represent the optic disc, okay? The blind spot. And that is the sheep eye. All right. Any questions, concerns, guys? Feel free to email me. Again, lab exam's coming up pretty soon. Okay, so make sure you guys are ready for it. It's actually next week, right? So be ready for that. Okay, so have a good day, have a good night, and I will see you guys later.